Okay, so today I'm going to show you guys the actual replication process of DNA. It takes a while, so I'm not going to give you all the notes on it today. We're pretty much going to draw a picture, and I think that'll take us to the end. But before we get to that, few vocabulary words I got to go over with you guys. So what we're doing today is we're learning what happens during interphase, and if we want to get real specific on it, we're talking about S phase when the DNA replicates. Okay, so that's our first bullet there. Second bullet going down, helicase. What we're going to do is we are going to unwind our DNA, and I'll explain to you why we do that later. But helicase is going to be the character that is going to break the hydrogen bonds between our two strands of DNA. So it's going to pretty much cut our DNA right up the middle. In cutting the DNA up the middle, it makes something called a replication fork. And don't think of like the silverware, like a fork like that. Think of it like a fork in the road. What it does is it forms like a Y shape in between our two DNAs, and you'll see that in the picture. Now, our goal is to make two identical strands of DNA. So what we have to do is we have to bring nucleotides over in order to make those new strands. Our next character there is called DNA polymerase, and that is the character that we're going to use in order to add the nucleotides so that we can build these new strands up. And again, you'll see that in the picture I'm going to make. Lastly, mutation. So sometimes we can have a mutation in our DNA. That's not good, and we'll talk about later on how we fix that mutation. So during the time of Watson and Crick, they had three different models about DNA. So the first one was the semi-conservative model. What this model was saying that, and I'll draw a picture of this as well for you, is that when we are making our DNA, we're going to make two cells in the end of it. So the semi-conservative model says that each one of our cells gets one of the original strands of DNA and one of the new strands of DNA. Other models were the conservative one, meaning one cell is going to get both of the original strands and the other one is going to get both copies. And the last one is dispersive. Dispersive is just like everything is everywhere. You're going to have some parts of the old, some parts of the new. But here's kind of what the picture looked like. Now, I'm going to draw it for you guys because I think you'll understand it a little better with the drawing. All right, so let's start off with the conservative model. So here is our two cells at the end of mitosis, and they would have nuclei in them, guys, but I'm not going to draw the nuclei just to save some space here. And we are going to say the red DNA here, that's our original DNA. And we're going to say the blue one is going to be our copy. So there's our two strands, guys. I'm not drawing all the nucleotides in the middle. The conservative model says this. One of our cells is going to have all the original. The other one is going to have our copies. All right? So that's the conservative. The semi-conservative still going to have our two cells here. This one says that each one of them is going to get a copy, and each one of them is going to get an original. Okay? So that's our semi-conservative. And then lastly, we have our dispersive model. Okay, in our dispersive model, guys, everything is literally everywhere. Okay, so there you go. There you go with the dispersive, guys. It looks a little bit crazy. What's going on here is we have fragments of the original and the copy in each of the strands, okay? So what they did, guys, they did a lot of experiments with these three to determine which one is the correct way that our ending cells after mitosis are going to have that DNA. And what they found was that the semi-conservative model, this one right here, this is our correct model. What they did is they used radioactive isotopes. I'm sorry, not radioactive. Isotopes, just heavy isotopes, in order to determine which one of these was correct. So they found that using the isotopes, that when the cells replicated, we had some of that isotope in each one of them. Okay, so that's how they found out it was a semi-conservative. Dispersive, we're probably going to have a lot of mutations in that one, so we don't want that. Okay, so throughout this process, guys, as I go through DNA replication, keep in mind that, we talked about the 5' the 3' end last time. The 5' end always has to be across from a 3' end. We can never have 5 across from a 5, or if we're talking about the same strand here, we can never have a 5 across from a 5 on this side. That does not work. It always has to be 5 across from 3. This would be 5 across from 3, and then same thing that way. Okay? All right. So let's get it. So this is going to be our DNA replication. And we are going to take this nice and slow. Okay. So here is going to be our original strands of DNA. Now, on this side, they're going to be all coiled up like so. I'll make the other one red. Okay. There's our 2 strands of DNA. I cannot draw them very well, but it's the best I got. So what we're going to do is we are going to open up that DNA. In other words, we're going to make something called a replication fork. And what we're going to do is I'm going to make it like that for the one strand. And for our other strand, I'm going to make it come up that way. Okay? So our first character we are going to talk about is this guy here. Okay. And that is Helicase. And what Helicase does is breaks the hydrogen bonds between our nitrogen bases. As he breaks them, so he's moving to the left of the screen. All right, so Helicase is moving that way. Kind of, I made him try and look like an arrow there, but he's going to be moving to the left. As he is breaking these bonds, he's also uncoiling our DNA. All right, so the other thing, he uncoils the DNA. In doing that, he makes this area right here, and that's going to be called the replication fork. Now, I don't know if you guys have ever done anything like this, but if you ever had like 2 pieces of string and they're all knotted together, and you're like, oh my goodness, I gotta like untangle all these. And instead of untangling them, you just pull them real tight. And when you pull them real tight, they just get knotted up even more. I don't know if you guys have ever seen that, but the same thing would happen with DNA over here. If we are having Helicase go to the left, right here, we are going to have this area over here to the left of Helicase. As we're pulling these strings apart, this area is going to get very, very, very tight. If it gets too tight, it will actually rip. So what we're going to do is I'm going to bring another guy over here. And he's going to be chilling on this side. And what he's going to do is make sure the DNA doesn't get too coiled up over here where it could possibly tear. So what he does is he pretty much twists and turns DNA just a little bit to make sure it's not going to tear and break. And his name is Topo Isomerase. So he's an enzyme, Topo Isomerase. Anytime you guys see the ASE at the end, you know it's an enzyme. All right, so Topo Isomerase, again, he just puts little fine-tuning turns onto our DNA to make sure it doesn't tear. All right, now let me label some different parts for you guys. We're going to say this part here is 5'. So that means this red side over here would be 3'. If this is 5', this would have to be 3', and that means over here would have to be 5'. Okay? So today, guys, I'm just going to work on the black strand. Okay, this is the easier of the two. And then next time we'll come back and do the other strand, the 5' strand, okay, or the red one. All right, so our goal is to make another strand to complement this guy here. All right, so we want to complement our 3' strand. So let me put some nitrogen bases in here for us. Okay, good enough. Now, if I were to do the red strand as well, and I'm not going to copy the red strand, like I said, we'll do that later, but we better have these matching up. So A would bond to T. T would bond to A. T would bond to A. G to C. C to G. T to A. G to C, C to G, A to T, T to A, and C to G. Okay, so with that, guys, our goal right now is going to be to make a full copy of our DNA using only the black strand, okay? And what we're going to do is we are going to make a complementary strand to this black strand, so that way we get a full copy of our DNA. Now, if you notice, like we just made, the red strand would bond right to the black strand here. Okay, so pretty much all we're doing today is making another red strand. So here's what we're going to do. We're always going to start on our new strand. We are always going to start on the 5' side. It always has to be that way, okay? We can never start on the 3' side. So I'm going to start, actually I'm going to draw a different color first. Let's go with a blue here. So our first few our first few nucleotides we're going to be putting in. Okay. Now, these first few strands, this is 5' here. Okay. This is called an Rna primer. All right. Now, what that RNA primer does is, and I know you guys are like, right now, come on, we're making DNA. Why do we got RNA in here, okay? Yeah, we should not have RNA in here. But we need RNA in order to make the DNA. So the RNA primer allows us for like a latching on point for the DNA to be made. So for RNA, now I have an A here. A, if you remember, it goes to T, but in RNA there is no T. There's actually a U instead. So A goes to U, and everything else is the same. T goes to A, T goes to A, and G goes to C. Okay? And we are going to be forming this right to left. So we're starting on the 5' and we are working our way to 3'. Okay? After this, we are going to get another character on here. Let me get a different color here. And right here, we're going to have this guy become attached. And his name is DNA Polymerase. And what DNA Polymerase is going to do is add nucleotides to the growing strand. Okay, so he's going to be moving this way, guys. So always from 5' to 3'. And as I'm putting these red nucleotides in, just so you know, he is the one that is putting them in. So I can't really animate him moving across, but he is moving across. So there'd be a G here. T would go to A. G would go to C. C would go to G. Now A, I know this part here was RNA. Now I'm on DNA. This area is DNA, so A would go to T once again. T would go to A. I'm running out of room here, but C would go to G. Okay? And what we would do, guys, in the end is we would have, because as we're moving this way, Helicase is also moving that way. So we're going to get more nucleotides coming out. And I do have another video that I will post on Canvas for you guys that actually shows the animation of Helicase moving to that way while DNA Polymerase is following Helicase and just adding nucleotides, okay? So that's kind of how things go, guys, with it. This strand right here, where DNA Polymerase is following the direction that Helicase is moving. Let me repeat that again. This strand where DNA Polymerase is moving the same way that Helicase is moving. They're both moving to the left. All right. That strand right there. So this copied strand is called our leading strand. Our leading strand of DNA. All right. We're going to make one down here next time. That's going to be called our lagging strand. All right. That one's a little bit more difficult. But I have an animation for you guys, another video. Please watch the video before next class, just so you can understand the leading strand really well. If you guys get that, it'll make the lagging strand a little bit more digestible. All right. Okay. So that's it today, guys. Just make sure you know, okay, so we got Topo Isomerase, puts the fine movements on our DNA so it doesn't tear. Helicase breaks those hydrogen bonds and also uncoils the DNA between our nucleotides. So if we wanted to say like this G up here and this C down here, it broke the hydrogen bonds in between them. All right. The RNA primer, that's how we start our DNA. We will take that out later, but it'll turn into DNA later. But we need the RNA primer in order for DNA polymerase to attach. And then lastly, DNA polymerase is going to add nucleotides. Okay. And let me add one more thing on that one. And DNA polymerase only can move 5' to 3'. Okay, that's important. So it only can move one direction. All right, it's like a one-way street. Okay, guys, so as long as you know that with the leading strand, I think we'll be okay for tomorrow. Just again, make sure you watch that other video animation that I post on Canvas. Have a good rest of your day.